First, you need to know a little about the histological structure of the gut wall. From inwards to outwards, it consists of the mucosa, the submucosa, the muscularis externa, with the inner longitudinal and outer circular layers, and on the outside you have the serosa or the adventitia. The motor functions of the gut are performed by the smooth muscle, and you need to know about two nerve plexuses. So the submucosal plexus present in the submucosa and the myenteric plexus between the inner circular and the outer longitudinal layers of the smooth muscle, and these two plexuses make up the enteric nervous system. The GIT smooth muscle acts as a syncytium. This is because the smooth muscle fibers are connected with one another through gap junctions, and this allows easy flow of ions from between the smooth muscle fibers. And secondly, the bundles within each layer, they are separated from each other by loose connective tissue, so even the bundles are connected with each other. So because the smooth muscle fibers are connected to each other and the bundles are also connected to each other, an action potential which is elicited at any point within the muscle mass will travel in all directions from that point. So that's why it is called a syncytium. So the electrical activity in GIT smooth muscle is of two types, the slow waves and the spike potentials. So the slow waves are oscillating changes in the resting membrane potential, and they occur spontaneously. The cause of these slow waves is basically they arise in the interstitial cells of Cajal, which are they are basically interposed between the smooth muscle fibers, and these interstitial cells act as pacemakers of the GIT smooth muscle. These slow waves are caused by the entry of sodium ions from to the sodium-calcium channels. The slow waves are not action potentials because they don't cause the entry of calcium into smooth muscle, and calcium is required. It's the entry of calcium that causes the action potentials, and once the threshold is reached, which is about minus 40 millivolts, the membrane potential becomes closer to the threshold potential. And when it does reach the threshold, you can observe many spike potentials, and these spike potentials are the actual action potentials, which are caused by the opening of the slow sodium-calcium channels. Another difference between slow waves and spike waves are slow waves are characteristic for each part of the GIT, which means that they stay constant for that specific part, but they aren't affected by nervous stimulation or hormones. However, action potentials are affected by nervous stimulation and hormones. The purpose of slow waves is to determine the frequency of contractions for each part of the GIT. For example, in the stomach, this frequency is 3 slow waves per minute, and in the duodenum, this is 12 slow waves per minute. So basically, the frequency of the slow waves is determining the rhythm of contraction, and this is different for each part of the GIT. The normal resting membrane potential in GIT smooth muscle is about minus 50 to minus 60 millivolts. Depolarization is caused whenever there is stretching of the smooth muscle, stimulation by acetylcholine, parasympathetic stimulation, some GIT hormones, and hyperpolarization occurs when there is sympathetic stimulation, so when norepinephrine and epinephrine are released. So now, what is meant by tonic contraction of the GIT smooth muscle? So this is the continuous partial state of contraction of the smooth muscle, and this is caused by repetitive spike potentials, some hormones that cause depolarization, and continuous entry of calcium ions, which has the same effect. The enteric nervous system is the intrinsic nervous system of the gut, extending from the esophagus to the anus, and it can function independently of the autonomic nervous system, but it does get input from the autonomic nervous system, which can enhance or inhibit its functions. It consists of 2 nerve plexuses, the myenteric plexus between the inner circular and outer longitudinal layers of smooth muscle, and the submucosal plexus present in the submucosa. The myenteric plexus is concerned mainly with controlling smooth muscle activity and GIT movements. For example, it determines muscle tone, and the velocity of conduction of the waves, the intensity of the contractions. But the submucosal plexus is concerned with mainly controlling local secretions and blood flow. It also receives afferents from the GIT epithelium, and these sensory signals are integrated in the submucosal plexus, which then carries out its own functions. Okay, so the autonomic nervous system can change the degree of activity of the enteric nervous system. So it receives preganglionic parasympathetic neurons because the postganglionic ones are present within the enteric nervous system, and it gets postganglionic sympathetic fibers from the prevertebral ganglia. So the parasympathetic nervous system, it consists of the craniosacral outflow. It consists of two things. So the vagus nerve, which will supply from the esophagus to the right 2/3 of the transverse colon, and the pelvic splanchnic nerves, which will supply the left 1/3 of the transverse colon and the remaining parts like the descending colon, the rectum, and the upper part of the anus. So there is some variation in the parasympathetic supply. The sympathetic nervous supply to the gut arises from the T5 to L2 segments of the spinal cord, and basically this gives rise to the greater, lesser, the least splanchnic nerves, and these preganglionic sympathetic fibers synapse in the prevertebral ganglia, like the celiac ganglion, the superior mesenteric, the inferior mesenteric, and from these ganglia, the postganglionic sympathetic fibers will supply the gut. Now, the difference between the functioning of the sympathetic and parasympathetic nervous systems for the gut is that parasympathetic stimulation is excitatory, which means that it will increase muscle tone in the gut wall, it will increase peristalsis, it will cause relaxation of the sphincters, and it will increase digestive secretions. But sympathetic stimulation has the opposite effect. It would decrease muscle tone in the gut wall, cause contraction of sphincters, and inhibit the release of digestive secretions.